Hey everyone, so here's the week 7, whatever week this is, week 7 lab, the last assignment. Thank you very much. Make sure that you read the manual through. You're also going to need to print out all these suckers so you can chop them up. And of course you'll be answering in your lab assistant. It says first message thread removed just because it was having a technical issue and didn't load the rubric post, so I did a new one and then it loaded it, and I decided deleting one was less confusing. So rubric, you have your rubric here for week 7 lab. You get your free S post for sharing your tables, of which there are 3, and of course the video playlist and the sample paper, because it makes me happy to keep giving it to you. Okay, let me pause this and open that rubric. Alright, so in this one, each exercise is 3.5, which would be 7, not 5. That is interesting. Let me pause that and make a couple corrections. Alright, now that I can add 3.5 for each exercise out of 7, makes 1 for format, which just makes sense. This is what happens when you sit there and try to do 1,000 rubrics in one day to get ready for a new class. Okay, so this one, the exercise one, only has 1 table. It's going to be 0.5. Each of those Punnett squares for a trait. So there's like potentially 2 for each trait. So if it's 1 trait and has 2, then both of them together would be 0.2 or 0.1 each, but some of them won't necessarily have 2. So yeah, so like, you know, dimples. If you do 2 Punnett squares, 0.2. If for some reason you only have to do 1, still 0.2. Okay? And then each question answer is 0.3. You'll see if you add this up, it's slightly less than 3.5. I think it becomes 3.4 because you just have a 1/10 of a point for turning something in. Sometimes I'm nice like that. Okay, and then exercise 2 is going to have 2 tables. Each table is worth a point, and then each question, there will be 2, is worth 0.75. Here we go. Format, 0.5. Citations, 0.5. You got to have at least 1. Okay. So let's go down and look at what this is. As always, just the copied and pasted assistant, or whatever they call it. It says here, assistant. And so you have a trait, your phenotypic characteristics, your phenotype and genotype of parent 1, of parent 2, and then potentially of offspring, right? So they're always going to give you parent 1. You yourself will be parent 2, and then offspring will be the result of your Punnett squared. Right? So it nicely tells you what the traits are. If it says just a capital letter, read the manual. It should tell you stuff. Okay, and then free blah blah. So you're going to look at temple, chin, free earlobe, ability to taste the PTC strip, interlocking fingers, what's on top, digital hair, the fun things we look at, the bent little finger, the widow's peak, hitchhiker's thumb, blue or green eyes, and rolled tongue. Okay, and then here it tells you about stuff. So like the dimpled chin. So you see here there's 2 for dimpled chin. You'll put in parent 1 what they gave you, and you'll be parent 2. And if you have a recessive, you will have only 1 option for parent 2. But if you have a dominant, there will be 2 options, right? So you do that for all our Punnett squares. There's a lot. Keep scrolling because there's a lot of traits. And then the questions will be, parent 2, how many recessive traits did you express? What were they? And how many dominant traits? What were they? And then while we're looking at these hypothetical genetic traits, what type of traits do you think are more important to study? Give an example of a genetic trait carefully monitored when 2 parents are creating offspring. Why is it important to monitor? So there's some traits where the dominant might be an issue or the recessive. And if you go over and look at your manual, I believe it covers some of that stuff. Okay? It's vague in my brain right now, but it has a whole list of disorders. And then we're going to have our karyotype image and what the disorder is. So this is another one where you take a picture of it and plug it in, right? Your disorder, your genotype, your phenotypes. You'll do that for all of these suckers. And then you will describe a karyotype and how it is performed. Sounds like you might need a reference. With a normal karyotype, meaning that a person would not have any disorder or disease in their life. Explain your answer incorporating the definition of genetics. So think about that. That's a fun one. Okay, so you get to cut and paste, pretend you're in kindergarten. It will be really fun. I love karyotyping. It's cool. And Punnett squares, lots of good practice, which is always nice, right? As always, submit a draft before Monday at noon. I'll happily review it. And, PM me if you have any questions. Share those tables to get some points, right? It might be tricky with the pictures, but we'll figure it out. It's a challenge. Why am I clicking on save? I don't know. I'm going to click on done because I'm done.